Vaccination is a hot topic, that we can't deny. And it has long been an effective way to reduce disease burden in humans as well as animals. According to Dr. Arshad Mather from the ARC, we can see it as an insurance policy. You're paying for something, but you don't see a benefit until there's an outbreak and your vaccinated animals survive. Today on Plaas TV, we focus solely on this relevant topic, so stay tuned while we see what's happening in the latest agricultural news.
In the news, the Supreme Court of Appeal has dismissed the National Council of SPCA's leave to appeal the Makanda High Court's ruling on an interim interdict. The interdict prohibited the company Al-Mawashi from exporting live sheep from South Africa to the Middle East. This is the sixth case the NSPCA lost at Magistrates High and Supreme Court levels in its bid to litigate for a ban on the transportation of live animals by sea. Let's hear what Arshad Mather from the Red Meat Producers Organisation has to say.
Dit baan dan die weg om die gedeelte B van die hofsaak op 'n stadium aan te hoor waar die Nasionale Dierebeskermingsvereniging vir 'n totale verbod vra en wat dit teengestaan sal word deur die regspan van die Rooi Vleisbedryfsforum. Ons is op rekord. Kom ons doen dit reg, dan kon ons dit vir altyd doen. En hoe doen ons dit reg? Ons moet 'n protokol in plek kry. En is die RPO saam met die lede van die Rooi Vleisbedryfsforum op rekord om te sê ons sal selfs die departement van landbou help om hierdie protokol te skryf. En ek dink hierdie hele proses is nou aan die gang. En hopelik sal ons by 'n antwoord kom waar daar 'n protokol in plek is binne die OIE se reëls waarop afgeteken kan word en dan kan hierdie handel op 'n permanente basis geskied.
And those were the words from Arshad Mather from the RPO. Two well-known agricultural companies, Villa Crop Protection and Intellichem Group, have merged under the American brand Winfield United. The combination of these companies in South Africa will create a platform for the development of an improved value proposition. This will include technology, scientific data and genetic seed provision. En professor Henny Snyman is onlangs as een van Stanford Universiteit in die VSA se top 2% navorsers ter wêreld aangewys. Snyman is 'n afgetrede hoogleraar van die departement Weidingkunde aan die Universiteit van die Vrystaat. Altesaam 165 van Snyman se publikasies het al in geakkrediteerde wetenskaplike joernale verskyn. Hy het ook 107 nasionale en 55 internasionale kongresbydraes gelewer. En dis dan vandag se nuus.
Netafim. Grow more with less.
A very relevant topic these days is that of vaccines, whether it's human or animals. As Dr. Arshad said, it's actually all the same. But we're going to talk about vaccines, how they are developed, what is the differences between a vaccine, a medication or a treatment, etc. So stay tuned throughout today's program. Welcome, Dr. Arshad Mather.
Yeah.
Welcome.
Thanks.
And he's from the Agricultural Research Council. So explain to us, what is a vaccine?
So a vaccine really is to simplify it, is sort of teaching your immune system to protect you. So in the use of a vaccine, it's a biological material which teaches your body how to protect itself against that pathogen or that disease-causing agent, the virus, the bacteria, the mycoplasma, whatever that agent is, that germ, like how we all refer to. And that's really what vaccines are. They're living biological material. Usually, you get two types of vaccines. They call them therapeutic vaccines, which is protection against this thing, and you get a treatment vaccine. They are very new. They're not really used much in the space of the veterinary sciences. The treatment vaccines, they're starting to pick up now in cancer treatments. So they give you a vaccine after you got cancer. Whereas the therapeutic vaccines is really protection. So you'd want to give that before an animal is sick and not when an animal is sick with a disease. And that's really what vaccines are in simple terms. Yeah.
So what's the difference between a vaccine and medication or treatment?
So a vaccine is given before the animal is sick to protect the animal. And if you want to, you can think of it as sort of an insurance policy. Okay, so your animal is not sick and you're giving it to a healthy animal. And it's really teaching the animal's immune system. So you're relying on the animal's own system. And this goes across to humans as well. It's teaching the organism how to protect itself when it sees this bad guy again in the future. Whereas a medication is when the host or the organism is sick, the animal is sick, and you're then treating the disease. And this is why we have this, first of all, reluctancy, because it's like an insurance policy. You're paying for something, the vaccine, and you don't see benefit. But you really see that benefit when there's an outbreak in that area and you see your animal surviving because you had vaccinated against a specific disease, as compared to your neighbor on the opposite farm who didn't vaccinate and his animals get sick and die because of that specific agent which is floating around in the environment.
So in very simple terms, how do vaccines prevent disease?
So vaccines prevent disease by giving a memory to the body. And all organisms have this way of protecting themselves, and it goes across all living species, plants as well, animals, humans, plants. So they have their own immune system to fight off pathogens, what we would term in science, which is germs really, So whether they be viruses, bacteria, etc. But what we're doing in the vaccine is we are giving the immune system of the organism a sort of a taste of this bad guy, really. Right. Saying, look here, this is a bad guy. Create a memory, remember him, so when he comes to take you in the future, yes, down the line, this is a bad guy. I need to quickly switch on my mechanisms, So you get a quicker response when you give a vaccine to an organism or a body, as compared to not giving a vaccine and relying just on the organism itself fighting that infection. So you're kind of creating a memory in the immune system of the host. So it remembers, yes, the next time it comes across this pathogen, this bad guy is like, yes, he's a bad guy, and I've seen him before, and I know what to do now. I need to switch on this mechanism in this pathway and this to protect me against this organism. Yeah. So that's really what we are doing when we vaccinate animals. And I guess, this concept is hard for people to understand because it's like, I'm healtgy,
Why do I need to go and get a vaccine? Can't my body fight?
Correct. Or even the animal. Why should I spend and, give my animals? They're healthy, they're walking around, But this is what you're doing. You're giving them that edge. So you're giving them a memory of a bad guy. So the next time they see it, they're going to elicit a better response. And usually, even in diseases which are fatal, diseases which cause death, in giving a vaccine, you get that edge where that disease, you may still get the disease, because we always sometimes hear people, farmers saying, I vaccinated my animals, but they still got sick, or, they're still feverish or whatever, but the thing is, you could have prevented death. Had you not vaccinated, you just can't tell. They may have gotten that fever, but also because they never seen this before, it's the first time they're fighting this virus, and then you get death because the immune system is overwhelmed with this pathogen, this bad guy. Yeah.
Well, there are different vaccines, so stay tuned. When we come back, we'll continue our discussion.
Goeiemôre, hierdie week kyk ons na die Mei-kontrak van Sonneblom en ons kan duidelik sien dat die prys steun gekry het by daardie tendenslyn wat wag by 9086, 'n loopie na bo verder ingesit het en nou is die groot vraag, gaan daardie hoogtepunt wat wag by 9240 verder in 'n steun verander? Met ander woorde, gaan die instrumente die momentum na bo voortsit? Ons kan aanneem dat die momentum wel na bo voortgesit sal word wanneer 9240 weer in 'n steun verander. Die eerste teken van swakheid gaan onder 9086 sigbaar wees en dit sal ook die volgende laer hoogtepunt bevestig wanneer die instrument wip, maar nie terug gaan na 9240 toe nie. Daarvandaan momentum verloor en wanneer 9086 in 'n weerstand verander, sal die volgende laer hoogte bevestig word of die omgekeerde koppie en handvatsel formasie wat kan impliseer dat die top van Sonneblom vir die Mei-kontrak in plek is. Neem ook kennis dat die rooi lyn verteenwoordig die 10-dag bewegende gemiddeld en hy wag by 9062. En dis 'n lekker gemiddeld vir korttermyn spekulante om te gebruik om te sê die korttermyn sentiment heens die instrument is positief solank as wat my 10-dae bewegende gemiddeld 'n steun bly, maar hy verander na negatief sodra die gemiddeld 'n weerstand word. Ons kan ook sien op die stochastiese indikator sit ons hierso op omtrent 'n 80 sone. Hy is oorgekoop, maar kyk, daardie twee lyne, daai blou en daai oranje lyn wys steeds opwaarts. So ons kan aanneem dat die afwyking, negatiewe afwyking bevestig sal word wanneer die instrument begin omdraai, 9086 in 'n weerstand verander en dan kan ons aanneem dat die momentum na onder kan voortduur. Moenie vergeet om volgende week se vrae weer vir ons deur te stuur nie. Geseënde week vir julle. Totsiens.
NutriFeeds, die formule vir sukses.
Welcome back to our discussion on vaccines. With me is Dr. Arshad Mather. He's from the ARC, and we are talking now about different vaccines. Yes, there are different types of vaccines.
Yes, and this is kind of a hot topic now because people are starting to understand the different types of vaccines. We talk of vectored vaccines, we talk of RNA, DNA vaccines. And so with regards to veterinary vaccines, same story, the history is we've spent hundreds of years with vaccines on our market, and they are all live attenuated vaccines, attenuated or inactivated vaccines. And just a little bit detail about that. So in a live vaccine is really a living organism, and an attenuated vaccine, which is really most of the veterinary vaccines are attenuated, and a lot of most of our human vaccines also, you really got the pathogen causing germ or virus, which you then have weakened. So you've weakened it in a specific way. And in attenuation, it's really a untargeted way. So what we do in the laboratories, we'd grow this, we'll find the virus out in the field from animals, or if it be humans, and we'd grow them in the laboratory over many, many years. And as time goes by, this virus itself starts losing specific genetic material and specific proteins on itself, because it's like, I'm growing in a petri dish, I don't need this gene anymore, because it's not giving me an advantage, making an animal sick, etc. So it throws out these genes randomly as time goes on over many, many years. And eventually you get this virus, which is now, it's got rid of some of its genetic material, which when you use it in an animal, it doesn't make the animal sick. But it gives that animal this memory. It's teaching that animal that, look, I'm so-and-so, but I can't make you sick anymore, And that's what we call an attenuated vaccine, really. So this process that we've been using for many years, and it's not a very scientific process as you've understood now, because you're just growing it up in the lab, in a petri dish, and you're hoping for the best. Like, okay, let me try. So you grow it up, maybe a year, 2 years, and then you inject an animal and you say, let me see if it's causing disease, and it's still making the animal sick. Let's go on a further growth period, So it's not very scientific per se. And the other one would be inactivated, and they also on a similar process, but in the inactivated process, you're really using chemicals.
Right, so it's not that the virus
itself. It still
itself
is the virus itself,
okay.
But you'll chuck in a few chemicals and say, okay, I got these chemicals, and we know these chemicals cause mutations. And again, it's sort of the same sort of bunch of chemicals that cause cancer in our body. All these chemotherapy agents and all these known drugs which cause cancer, because they're disrupting DNA and disrupting at that level, at that molecular level. So you chuck them in with a virus, and it disrupts some genes. And again, we hope that it will be the genes which make the animal sick. And you test them, and those are what we know as inactivated vaccines. And so you got those two classes. So they're living, both of them, they're living viruses which have been weakened by a specific process, either culturing in the lab over periods or with chemical disruption. And that then makes a vaccine. So the body still sees this virus, but it's not so bad anymore. It's not such a bad guy anymore, because the virus threw away some of its genes. Then a more recent process, and these are what we now call new generation vaccines, is where we look at a scientific way of doing this. So, scientists started saying, this process, we don't know what genes are being disrupted and what's being chucked out. Let's look at the DNA, since we now can sequence and we can look at that molecular level, that DNA which makes up the whole virus itself. Let's look at it and see which genes are involved in just the virus growing and its daily functions of energy and survival, and which are the ones making the host sick, the animal, the person sick, And when we find out those genes, we use those genes then to make the vaccine. So, and I'll use the hot topic now, which is in the SARS research, right? So they identified the spike protein, that, oh yeah, this virus is using the spike protein to enter the cells in the body. So, let's find out that DNA that codes for it. And DNA is kind of a code, a sequence of nucleotides which make proteins, right? So we found out, okay, we know the spike protein is an entry. So let's now make a vaccine with a spike protein. So the body recognizes that spike protein, and then when the virus as a whole comes in to make an organism sick, it's got antibodies, it's got memory against that spike, and says, yeah, this is a dangerous guy, this spike thing. Let's make protection. Let's kick on that mechanism of immunity, And that's really now where we talk about subunit vaccines. So subunit is not the living whole virus. It's just that one protein from the virus. And it's either now vectored, which is now, like as an example, the Johnson & Johnson vaccine. And then for my research, I'm specifically doing a vectored lumpy skin vaccine. That's what I'm looking at, and Rift Valley fever vaccine. So you'd identify now those proteins which are disease causing or kick switches on your immune system in the host, and you're just taking that one protein and you're putting it into a carrier, and in a vector, it's kind of a Trojan horse.
Right.
So you hooking up this protein into a Trojan horse, which goes into the immune system and says, yeah, so the Trojan horse is not doing any harm. That's the vector, not doing really any harm, but showing the immune system, look, this is the spike protein, this is the Rift Valley fever protein, this is the lumpy skin protein. Kick on your mechanism and remember this the next time this virus comes, And those are then the vectored vaccines that we talk about here.
We have to move on. A question I think we all have, seeing we are in the COVID times. Are vaccines safe?
Yes. They are safe. And I say that because of all the regulatory mechanisms and the research that goes into really vaccine development. I mean, these are really long processes. And before a vaccine is really marketed, you've got all these processes in place. No doubt you do see side effects, you do see, and as a researcher, we say that's not bad really, because it means the body is recognizing something,
right?
And this is why, like and again, in this Johnson & Johnson, it's a bit hot topic. In lumpy skin as a vaccine, also we hear this. At the site of injection, so you see the swelling, and you see sometimes pain in that limb or that arm. You get fever, and these are, again, like I say, because we're teaching the immune system to recognize the bad guy, right? So the vaccine is really doing its job. It's teaching the system.
So you see a reaction, and that's what you want.
And those reactions are the immune system's reactions. It's a normal reaction to get rid of a virus. When a fever, viruses don't like hot temperatures, so the body raises the temperature and says, let's warm up my cells a little bit so the virus can die, because they don't like hot temperatures, and that's your fever reaction. And then the swelling is inflammation. Those drugs which cause the swelling is really bringing a lot of blood flow to that area. It's swelling cells. It's creating this environment, these chemicals in that area that the virus doesn't like, to try to get rid of it. So it's an expected response. Obviously, we don't want to do it all the time, because people don't like that. They see the animals and it's like, is my animal getting really in pain because of that swelling? So that's why we minimize those post-vaccinal reactions, those reactions at the vaccine site.
Well, we can talk about this topic for another 3 days, I think, but we're going to take a short break, and we'll be back with our discussion. Welcome back to our discussion with Dr. Arshad Mather from the ARC, and we are talking about vaccines. Yes, Doctor, how is a vaccine developed? Explain to us shortly, briefly, the research and the process.
Okay, so vaccine development is really a lengthy process, even the newer generation vaccines. And I mean, to summarize, maybe up to between 10 and 15 years before you really see a product on the market. So the first stage, which is usually about, 2 to 4 years, would really be the development phase. And this is where you're identifying those bad guys, those bad proteins on the virus, or the many proteins on the virus. You're understanding the virus itself, or the bacteria, the germ. You're really learning about the germ, identifying which genes you want to use, where and how. And then the next phase is animal testing. And this usually takes between 1 and 2 years. And this is where now you vaccinate that animal with your construct or your vaccine, your potential vaccine. You try to identify things like, what is the dose I'm going to use for this vaccine? What is a maximum dose before the animal will get too sick? Remember, because some of these things do make you sick at high doses, So you decide doses, you decide whether it's safe in pregnant animals, etc. All those sort of results you'll get. And then the next stage after that is really, once you've got that information, that research data, you take this onto a field test. So usually in a laboratory test, there's a small number of animals. We're talking, 5, 10 animals where you'll vaccinate, and then you'll challenge. Challenge meaning give them the agent itself, the disease-causing agent, and you see, does my vaccine work, And then in the field trial, you really want to go up to about 100 animals on a farm setting. And this animal research takes place over a year and a half. And you're looking at a field infection. So really, the animals are in the field, you vaccinate them, and you're looking for a natural infection. Because again, in the laboratory, we can't completely replicate what happens in the field. In the field, animal sees a lot of other viruses. Its immune system is not this clean immune system.
And it's temperature, and it's water.
Correct, environment, food is different. In our lab setting, it's all control, that's what we want to do. But in the control setting, we see our vaccine works. Yes, okay, now let's take it in this uncontrolled, multivariable setting, and that's in the field, And then you do that, and then your next step is really looking for registration with our authority. And people all know now about the authority, SAPRA, the South African Health Products Regulatory Authority. And you'd put in an application. You'd say, look, here's my research data with all the tests. Here's my animal data, all the results. This is what happened. This is how the organism responded. This is what the virus did. This is what the vaccine did. Here's the field data. And that process takes also between a year and two for these people to sit and evaluate those results. And then come back to you and say, no, we're not happy. This test, you did too few animals. Statistically, you need more animals. And once all this is done, SAPRA would give you then that authority and say, okay, you can now manufacture and sell this product. And then that's when you'd partner. So ARC doesn't do manufacturing and marketing on its products, except the FMD, which we make ourselves. The others we'd partner with the other state-owned entity, in most cases, which is OBP, Honest Report Biological Products, where we'd hand over or in partnership with them the technology, and they then bulk it up or scale up. That's the word we use in sites, where they now say, okay, in a small lab, in a small dish, you made so much vaccine. Now let's make that in a larger amount. Let's do the quality testing for that. And that process also is now you get a better idea why I say vaccines are safe, because look at the number of years spent on this product. So by the time you really hit the market, you know a lot about it. You can never say, you're 100% sure about anything. Life's not like that, right? There's always that bit of uncertainty. So that is always there. But these products have been, so much research goes into them, and so much thought processes and experimental data and quality assurance that at the end point, you kind of got a safe product that you market, effective and safe product. Yeah.
So why is storing at a specific temperature so important?
So this is because, I told you, vaccines are biological materials. And biological means they're living, in a sense, in inverted commas. So you need to keep them at the temperature that will prevent them from degrading. So if it's a vectored vaccine, because the vector is live, the antigen or the pieces from the virus are not, but the vector itself, the carrier, the Trojan horse is live. So you want to keep it at a certain temperature. And we do research and see, okay, a good temperature is 4 degrees, So at that point, that components don't fall apart. And then mRNA, DNA vaccines have a different storage temperature. And this is why you got different storage temperature. And this is why you need to store them at that appropriate temperature. If you do not, you're degrading or destroying that biological material which makes up the vaccine. And in so doing, you're losing efficacy. So your vaccine won't be as effective. And that is a big challenge. And sometimes won't even protect at all. And this is then when we get complaints coming back from the field or farmers or people, like, you know what, I vaccinated, I spend money, it's not working, But did you store it correctly? You may have not stored it correctly and that living material degraded or got destroyed. So you thought you're giving a working vaccine, but you're giving a kind of destroyed vaccine, and then you don't get protection. And this is then why you think it's vaccine failure, but really it's not vaccine failure, it's inappropriate storage, inappropriate usage in the field. And these are challenges that us as scientists would now try to overcome, That is why we'd say, okay, let's do a vector because we know this vector is very stable at room temperature. One of the research projects I'm doing is like that. The vector is stable at room temperature, so you'd say, okay, let's use this as a vector for the next generation of vaccines because this vector is going to make that vaccine room temperature usable.
We have to say goodbye, but very briefly, what research takes place at OVR?
So at OVR, we across the spectrum. We're looking at diagnostic development, new diagnostic kits, new diagnostic tools development, and vaccine development for diseases across the veterinary diseases, specifically veterinary diseases. Things like Rift Valley fever, lumpy skin disease, foot and mouth disease, rabies, avian influenza. We cover the entire spectrum. And in fact, we're also starting to get into the COVID-19 research, and this is specifically with, does COVID infect animals? Can one animal give another animal, make another animal sick against COVID? Can a person get sick from COVID? We are starting to look into that also. And again, because we have the skill, we have the knowledge, we have the facilities at our institute, we can do like high level stuff in, BSL-3 laboratories where you're working with high pathogen, high risk viruses. Yeah.
Well, we'd like to thank you for what you're doing for our agriculture in South Africa, for the farmers. Good luck with what you're doing. And I think it's a very relevant topic with us also being very much aware of vaccines. Dr. Arshad Mather, thank you very much.
Thanks for having me.
And that's our discussion on vaccines for today. If you missed the beginning, go and have a look at it again and follow us.